Guys, let's start with the step 5. In this step, glyceraldehyde 3-phosphate is being converted into 1,3-biphosphoglycerate. That means a phosphate is added. Plus, I have written these two on each side because two reactions are occurring. We know that the 6C compound, that was glucose, is finally been broken into two 3-carbon compound. Those were glyceraldehyde 3-phosphate. So now with every reaction there will be two. That means that two reactions, one for each glyceraldehyde 3-phosphate is occurring. So now this is of course an oxidation reaction. The enzyme involved is glyceraldehyde 3-phosphate dehydrogenase. Dehydrogenase is actually a subtype of oxidation enzymes. This is an oxidation reaction and NAD is consumed. We can see here that 2 NAD enter into the reaction. Glyceraldehyde 3-phosphate dehydrogenase caused glyceraldehyde 3-phosphate to be converted into 1,3-biphosphoglycerate plus and 2 NAD to be converted into NADH plus H. What is NADH plus H? It is actually reducing equivalent, that is ATP source. Reducing equivalents are those compounds, which carry energy to electron transport chain, and when they go to electron transport chain they cause energy, that is ATP, to be formed. That is an entirely different respiratory chain, and that is not our concern right now. So aldehyde, in this reaction an aldehyde group is converted into carboxylase group, that is the conversion of glyceraldehyde to glycerate. That is the main change. A phosphate is added, just like I told you. Now the question, most important question of comes, where did the phosphate come from? In previous reactions, when a phosphate was added, it came from ATP, energy was expended. But in this reaction, it does not come from ATP. So where does that phosphate come from? It actually, we have to understand the structure of enzyme to know actually where does that phosphate come from. We know glyceraldehyde 3-phosphate dehydrogenase enzyme is actually a tetramer. Each of its monomer has a thiol group. One thiol group is actually at the active site. At the active site, this thiol group binds with the substrate, that is glyceraldehyde 3-phosphate. Now this enzyme has three pockets. One for substrate. I told you there was a thiol group which bound with that substrate. One for NAD, this NAD, and one for phosphate ion. So actually this question was, where did that phosphate ion came from? Well, the answer is that that phosphate ion is actually already connected to the enzyme, so this question should be no more. So well, let me explain it to you. Actually enzyme has three pockets, right? One for substrate, one for NAD, one for phosphate ion. All of these three things, when connected to these three pockets, then the reaction occur, and finally 1,3-biphosphoglycerate is formed, NADH is formed, and phosphate ion is added into this. What is the mechanism of this reaction? Very simple. When glyceraldehyde 3-phosphate binds to SH on the active side, that is thiol group on the active side, it forms a compound called thiol hemiacetal. Let me zoom that in. Thiol hemiacetal. Well, the two other reactions, that is thiol hemiacetal, will lose its 2H to NAD, that is 2 NADH plus H will be formed, and now the compound will be called thioester. The thioester compound will be phosphorylated and release a free thiol group and 1,3-BPG. This is a mechanism of conversion of this compound inside this enzyme, that was glyceraldehyde 3-phosphate dehydrogenase. We know this reaction is reversible as the arrows suggest, so it is not of regulatory importance, but it is of clinical importance. Well, the clinical importance of it is actually arsenic poisoning. Arsenic poisoning, we know that humans are poisonous, humans are actually vulnerable to arsenic. Arsenic, actually, what arsenic does is actually it halts the process of glycolysis, and ATP production does not occur in the glycolysis. So this actually, arsenic affects this enzyme glyceraldehyde 3-phosphate dehydrogenase, and also another enzyme which is called pyruvate dehydrogenase. Arsenic poisoning is mainly due to the effect on pyruvate dehydrogenase, but arsenic also affects glyceraldehyde 3 phosphate dehydrogenase. What actually does, it actually arsenic binds with the pocket for phosphate. This pocket right here, phosphate ion pocket, here instead of binding a phosphate ion, arsenic binds. As a result, the alternate pathway occurs and glycolysis skips our energy releasing step. So as a result, due to arsenic poisoning, the energy gain from the glycolysis is turned to zero. This is actually arsenic poisoning. Let's proceed to step number 6, which is also not an important step. Step number 6, 7, and 8. These steps are not important because these are reversible steps, and they are not of regulatory importance. Let's proceed. Now the first step number 6 reaction is 1,3 biphosphoglycerate is converted into 3-phosphoglycerate. We know as the reversible reaction arrow suggested, a phosphate, as we can see, is removed. A phosphate is removed. Enzyme is actually phosphoglycerate kinase. Phosphoglycerate kinase. Name suggests that kinase is actually transferring a phosphate from 1,3 biphosphoglycerate and transferring it to, well, the answer is ATP. ATP. 1,3-biphosphoglycerate, one phosphate from 1,3-biphosphoglycerate is removed by this enzyme and given to ADP, forming ATP. As a result, 3-phosphoglycerate is formed. This reaction is reversible, hence is of no importance, but this reaction, although is the energy gain or energy harvesting step, one of the energy harvesting step of the glycolysis. Here there are two ATPs gained because two reactions are occurring simultaneously, as then this 2 suggests. Step 7, 3-phosphoglycerate is converted to 2-phosphoglycerate. Here is the reaction. We can see a phosphate from carbon number 3 is moved to carbon number 2. The enzyme is actually mutase phosphoglycerate mutase. A mutation is occurred, that is a phosphate from carbon number 3 is being moved to carbon number 2. What is importance is actually the enzyme is phosphoglycerate 2 mutase enzyme. This is an isomerization reaction, and it is most likely that 2,3-PPG, that is 2,3-biphosphoglycerate, is actually an intermediate in this reaction. This is a reversible reaction, so it is of no importance. Step number 8, 2-phosphoglycerate is converted to phosphoenol pyruvate. Phosphoenol pyruvate. Well, the enzyme involved is enolase. Enolase enzyme converting 2-phosphoglycerate to phosphoenol pyruvate. This is a dehydration reaction. That is, hot water is actually removed from 2-phosphoglycerate to convert it into phosphoenol pyruvate. Although it is not clear that phosphoenol pyruvate has phosphate in it, but actually this compound has a phosphate in it. The name does not suggest it, but this enzyme, the phosphate didn't go anywhere. It is a reversible reaction, so it is of no importance. So now enolase, this enzyme enolase, this is of importance since this is a glycolytic enzyme. This is important. It is inhibited by fluoride. Suppose we take a blood sample and we put in some fluoride in it. What will happen? The RBCs, they get the energy from glycolysis, right? If enolase is inhibited, how will they get their energy? Glycolysis will not occur. So that is why we add fluoride to blood samples in which we want RBC to not run their glycolysis. That is when we take blood samples for glucose concentration in the blood, so that the results are not altered. So step 9 is another important step, another energy harvesting step, plus another irreversible step, has a major regulatory point, in which phosphoenol pyruvate is converted into pyruvate as the reaction I just wrote down. This is irreversible, arrow suggests. Enzyme is pyruvate kinase. Kinase, another kinase means transfer of phosphate. I already told you that phosphoenol pyruvate has one phosphate in it. So pyruvate kinase actually removed that phosphate and added it to ADP. and ADP was converted into ATP. Hence, pyruvate, the compound now formed, is not phosphate. It does not have any phosphate anymore. And this is the end product of glycolysis. Two pyruvate, one from one glucose, two pyruvate were formed. This is the final step, but this step is irreversible, right? This step is irreversible, hence of regulatory importance. Now, the fructose 1,6 biphosphate regulates it positively by feed-forward mechanism. This is a regulatory step, right? Now, fructose 1,6 biphosphate was in the very start, step number, I guess, 4 of glycolysis, in which fructose 1,6 biphosphate was formed. Fructose 1,6 biphosphate, a 6 carbon compound at that time, was formed. When that compound is formed, it, by a feed-forward mechanism, activates pyruvate kinase. That is, if increased glycolysis is occurring, right, increased phosphate 1,6 biphosphate will be formed. Increased phosphate for fructose 1,6 biphosphate when formed will cause the pyruvate kinase to increase its activity, and glycolysis will occur in a orderly fashion. So also hormonal control is there. That is, if the glucose concentration in blood decreases, then glucagon, that is a hormone, its concentration increase. As we already told you that increased glucagon concentration cause intracellular signaling and cause protein kinases to be formed. As a result, phosphorylation of pyruvate kinase occur, and pyruvate kinase is inhibited. Pyruvate kinase inhibited, hence glycolysis will not occur. Why glucagon was released in the first step? Because glucose was low. Body didn't want to spend any glucose, didn't want any glycolysis to occur. Hence, glucagon, by a by a series of steps, caused pyruvate kinase to be inhibited. This is step 9, another very important step, a regulatory step. We completed the glycolysis. We converted glucose into 2 molecules of pyruvate. Hence, now we can take a look at these all steps. These are all steps of glycolysis, right, in an orderly fashion, from glucose to 2 molecules of pyruvate. Let me zoom that in for you. We in the start gave 2 ATP. Expenditure of 2 ATP took place, but in the result we got 2 NADH reducing equivalents, which will give us energy in electron transport chain. 2 ATP, that is another energy gain, and another 2 ATP. These ATPs are actually being formed by substrate level phosphorylation. When reactions occur and ATPs are produced directly, that is substrate level phosphorylation. But when ATPs are produced indirectly by reducing equivalents, that is NADH, that is actually called electron transport chain oxidative phosphorylation. So we can now divide the glycolysis in 2 stages. In 2 stages. Number 1 is actually energy expenditure state. Here we can see the first is energy expenditure state. 2 ATPs were added into glycolysis. Energy was expended. The other is energy harvesting stage, in which we gained 4 ATP and 2 NADH. Now we can summarize glycolysis by its net equation: 1 glucose plus 2 NAD. Here there is NAD. I don't know why there is question mark written here. Here glucose plus 2 NAD will result in formation of 2 pyruvate, 2 ATP plus NADH plus H. Here there also should be 2 NADH will be formed. Net ATP gain, our substrate level phosphorylation, we get 4 ATP. At substrate level phosphorylation, that is these two directly gain of ATP, is actually we get 4 ATP, and respiratory chain oxidation of NADH will result into 5 ATP formation. Hence, the overall ATP gain was 9, but we had expended 2 ATP here, so the net gain, ATP expense was 2 ATP. So net gain will be 7 ATP. This net gain is very much very popularly asked by examiners. So that is all about glycolysis. In the next part of my video, I will tell you about the special cases in glycolysis when the reactions deviate from their normal pathways, like in RBCs, in the formation of 2,3-BPG. I will tell you about that, and also in different other things.